we talked about yesterday, often what you see is this knowledge pneumatic area is dispersed. And what you see here is often it's an active turbulence. Okay, so what happens in that case is you see, if you look at this picture here, you do not have long-range pneumatic order anymore. What you have is very localized ordering, and on the large scales, you just see a chaotic flow pattern. So this is the system of the microtubule motor protein system that we're specifically trying to model. Okay, again, you have microtubules. These are about micron sized in length. And when you put them in a solution of motor proteins, they're going to be cross-linked, and then they form these dense networks. And then you can observe these systems by putting them on the interface. So then all your water interface is a planar, which is for easy observation. And we talked about how do we derive the equations at the mean field level. So essentially, the main point is that it's not so different from what we've been talking about for the polar particles, except that these guys are not only generating stress and they're not self-propelled in that sense. So we talk about this equation, which is not different from the passive pneumatic equation, okay? So this will be essentially the evolution of the pneumatic order. And in terms of the hydrodynamics, that's essentially the same as before. So you have a Stokes flow kind of behavior, except that now, previously, from the polar case, we have the viscous stress, okay, just from the solvent. We had an active stress here, which is proportional to the pneumatic order, because we have elastic stresses here as well due to the fibers here. So we have that term. And if you compare this to a passive pneumatic system, this is the only new term that we have here. But everything that you see that's interesting is driven by this term here. Okay, so the point is it's not so different in terms of the equations. We just need to add that term there. Sigma is the total stress tensor. Yeah. So, and we also talk about how, specifically in the yeah.
That last term, the smile.
Which one? That's the active. What people often think about is to remove the elastic part, just have the viscous stress and the active part, and then look at how that is coupled to the pneumatic to the evolution of the pneumatic field. And then that's the simplest theory for active pneumatics that people have been thinking about over the years. And as we've seen before in the first lecture, if you look at these systems in an experimental setting, what you see is when there's no boundaries, just in bulk free space, if you do a periodic box in a simulation where you observe this experimentally far from boundaries, okay, you can do that. Level of activity. And then if you do an experiment, so this is a very dense system, and if you look at the structure of the flow, again, it's very chaotic, and you have these defects that develop in a system. And this is a simulation that resolves the PD system we talked about in the previous slide. So you are sort of be able to capture these structures in a simulation. So here's a video of the same system that we looked at in an interface. So it's planar. It's easy for visualization. What's interesting here, as you would imagine, because these are active, right? So the defect here is moving and evolving. Sometimes there will be it's on an interface. Yeah, that's easier to visualize. Planar. Yeah.
So that white part is the
white part.
the white dot over there. Is
Let's watch white dot. I'm not sure.
you said the
Uh, so these arrows, those are defects. Yeah. How they move around, it's also dynamic in the system.
is it an example of the
Pneumatics, they're generating forces. And what you can do is you can now, because of the friction, you want to add an additional term to your momentum equation to include the friction. That's all you need as a first model. So what you can do is to add your stress
that the equation that we had before is equal to 0, right? And you just add a frictional force to the equation, which is essentially body force per volume in terms of the dimensions. So you add a frictional force. And as a model for a flow without inertia, that will be linearly proportional to your fluid velocity, okay? And here, gamma, this would be a friction coefficient. So these kind of models are also widely used in the context not of vacuum matter, but of fluids in general. For example, if you think about fluid motion through a porous medium, where you have pillars arranged in space, and you want fluid flow through them, because you have to pass through all these solid surfaces, that the flow will be different compared to moving without these pillars, right? So you have additional friction associated with that. So that's also how you do that, and that equation has a name. It's called a Brinkmann equation in the context of fluid mechanics. And in applications, you often average these equations. For example, in the context of porous medium, you do an average over these pillars, and you can get a simple law called Darcy's law that tells you how fluid moves and what's the pressure gradient you needed to drive that flow. But in this context, we just want to think about what happens when we add the friction to the pneumatic. And when we add this, we can look at our equations. So if we take our equation here, momentum equation, in this equation, we have a viscous term, right? And we have the active term. And the friction. So if you look at these two terms, the first one is the viscous term from the fluid, and the other is the friction. So if you compare these terms, you're going to introduce a new parameter, now a length scale here. Okay, so let's define a length scale L, but by just balancing these terms. So on the left-hand side, we have the viscosity. We have two gradients, right? So that's 1 over the length scale squared. And we have the velocity, which we can drop out because on the right-hand side, we also have a velocity. So then if we balance these terms, we can derive a length scale L, which is given by the square root of gamma, or square root of mu over gamma, right? So we introduce a new frictional length scale to the system. So that's essentially a dissipation length scale due to the substrate. And if you increase the gamma here, you increase the frictional coefficient, what happens to the length scale? That's going to be reduced, right? So that's a parameter you can tune to control the turbulent behavior in the system. And let's now go back to the slides and look at what happens if we incorporate this kind of friction. Any questions about the derivation that we just did? Okay, so here's a simulation of these equations. And at the top, that's just the case without the friction. And you see the chaotic flow pattern that we've seen before. And what you do here from the top to the bottom and then to the right is to increase this gamma. Then you're going to decrease this frictional length scale. And as you increase gamma, what you see is the chaotic flow pattern is now suppressed. And in a large friction limit that's here on the right, what you see is how you have these much more regular flow patterns that are developed. So essentially, the frictional length scale is very small. So all these, for the turbulence, you have these length scales associated with the energy dissipation. So essentially now the energy dissipation is a much smaller length scale. So you can't develop this large chaotic flow here anymore. And the other thing is here, notice that also the defects have gone away, and you just have a very regular, essentially only aligned in one direction. So the defects are also gone. So that's one way to control the chaotic behavior in these systems, which is just by introducing a boundary, which gives rise to friction at the surface. Are there any comments about this? I'm not sure. But if you, again, go back to what we talked about before, right? We've seen this pattern already, even for the case of polar suspensions. When you confine them, whenever you introduce a new length scale, that will compete with what's happening in the system. So in general, yes, but as to the details of how it evolves, then that's something you have to reason about in more detail, yes.
Good.
Okay, so here's the experiment. So the one we've seen previously is a simulation. So here are some experiments. I believe these are confined between two plates, and then different width between the two plates. So here are some videos of the flow pattern that we're seeing here. Okay, so that's in a channel kind of environment. Yeah, you have this character as a shear flow kind of character, but you see it sort of switches back and forth sometimes, right? So the point is that trying to make this kind of flow very regular, say unidirectional, that's a non-trivial thing to achieve. So that's also the things that people want to understand. How do we control this kind of turbulent behavior? You know, if you want to use this flow for some application, for example, you want to be able to control where it's going, right? And
Is the kind of
yeah, so you can say you use these active guys to generate flow to transport passive things. Then you have to be able to control the flow, and then you have to be able to tell me if I want the flow to go to the right, you have to tell me what parameters I need to use and things like that. So those we don't know yet, and that's something people want to understand in terms of future challenges in these systems. Yeah, good. Question? Okay. So defects, as Vasker was talking about, so defects are everywhere in nature. For example, here in biology, you have a lot of cases where you have defects. For example, in this bacterial colony, whenever you have, so essentially, if you think about a colony that's developing, they're developing in all different locations, and locally they try to first achieve local ordering, but there's another region where they're also trying to achieve their local ordering, and these ordering are not necessarily aligned. So as they grow into one colony, there's frustrations that's going on here, and you naturally would have a defect. That's a bacterial colony of E. coli, and these are just epithelial cell tissue. You also have those kind of defects here, again, due to development. And here are other cases, stem cells, and this is again a fibroblast cells that you see these defects occurring. Okay, so those are active pneumatic kind of systems. And let's now think about a slightly different system of active particles, which are active droplets. Okay, so here's an experimental video.
Pneumatics, this the active pneumatics, what's happening to those people are, what are the properties which are interesting you in such as studies?
So largely, at the current stage, is just trying to experimentally design systems to understand active physics. And in terms of the application, right now there's not much. And previous work largely trying to understand essentially sort of the structure of the turbulent flow and the connection. The question is, because this is not actual turbulence that we're familiar with, and what's the difference between this kind of flow and the classical turbulent flow? You have a high Reynolds number, so that's the main thing that people are looking at.
In these cases, it says if that is costly, has
People haven't done this at all. Even as you said, in passive pneumatic system, trying to model the defect dynamics is quite tricky. Of course, people have done it, but to certain degrees. So it's an interesting but tricky modeling process that one have to do. Yeah. Great. Other comments? Okay, so we'll look at one last system that's common out there, especially in the last couple of years, that people have done is active droplets. So it's a droplet, some kind of liquid, put in another solvent, and then the droplet is gonna self-propel in some fashion. So here's an experiment I'm going to explain later on what kind of droplets we have here. But if you watch the movement, so this is in a spherical cavity, looks like active Brownian particles, right? They're swimming around. We did simulations yesterday. This is the kind of trajectories that you're getting. And here's a experimental image of the droplet that's moving around. And this is a zoomed out view of each different trajectories of these droplets. So there are two common types of droplets that one can use in these systems. One is a reacting droplet. So what happens here is you have a droplet, say spherical, right? And you have surfactants here that are on the surface. And notice you also have these micelles on the outside. And there are reactions happening in the droplet, which generates these red chemicals, these red, I don't know if you guys can see that, the red points here. So the red chemical will modify the surface tension behavior of the surfactant. So once you have a red gets here, we just color the black, which is the pristine surfactants, to red. So that changes the surface tension of the surface. And then what happens here is then actually if you do a linear stability analysis of the system, you can establish spontaneous flow. And when you have a spontaneous flow, when the flow is biased, and then you will advect these surfactants to the back, and then you further sort of enhance the surface tension gradient, which drives flow inside, and through force balances through the interface, you can sustain a motion of this kind of system. So it's a hydrodynamic, it's complicated chemohydrodynamic interaction that gives you this kind of behavior in a droplet. So this is one case. So you don't have to have these reacting species that's going on, because we just need to establish a instability where you create a surface tension gradient on the surface, which is balanced by the force stress difference inside and outside. So that will create motion. The other one is a simpler system. It's a solubilizing droplet, such as an oil droplet, okay, in water. And there are surfactants there. So what happens is the oil is going to be taken up by the micelles here outside. So essentially the droplet is dissolving, slowly dissolving, okay? And that's called micellar solubilization. So what happens is the oil slowly and continuously leave the droplet surface, and then you will produce a concentration gradient of these filled up micelles. And again, if you calculate to the hydrodynamic analysis, again, there's an instability that occurs. There's a basic state which is symmetrically distributed. There's no motion at all. And then when you have a stability develops, again, the flow migrates these guys to the left, and you induce a gradient that drives up their motion. So that's sort of active droplets and how often people use them to generate motion in this context. So again, we can develop a simple theory to describe it, a hydrodynamic model for it. So now we have sort of a new thing that's some kind of chemical species that comes out of the droplet into the bulk fluid. And that species follows a simple advection diffusion equation. So this UO, that's the flow field outside the droplet. And now we're experts about these equations. So those are just Stokes equations outside in water. And in the droplet, it's again a liquid, follows the same kind of equations, except those are labeled by I. Okay, there's some interfacial dynamics that connects the outside to the inside. One is we're assuming that the droplet just generates a constant flux of this chemical species, and then it comes out, and then it moves around by following advection diffusion dynamics. So that's what this term is saying. Okay, a constant flux A. And there are other mechanical balances. So the first one is there's a stress balance on the surface. So there's the stress outside, stress inside is balanced by the gradient of the concentration, which is drives the surface tension gradient. So these two terms are essentially just force balances at the interface that you have to derive. And this is the velocity difference here because of the chemical concentration that's present. So there are some phenomenological coefficients here. One is this A, that's essentially how much flux I'm getting. And one is this M, so you can view that as a mobility, which relates the sort of the slip velocity difference to the chemical concentration gradient that's present. So in an experiment, you can imagine if you solve these equations, you can try to fit these two parameters to your particular experiment, and then can make use of the model to describe what happens to the system. Okay. So now the final thing is we're not done yet with these equations. So this droplet is moving in a solvent. We're not applying any external force on them. So which means the droplet is force free. So then the total force is 0, as we talked about for other kinds of self-propelled swimmers or particles. Any comments about these equations? So we're not going to derive these here, but hopefully most of them we've seen already. So they are at least conceptually straightforward. Okay, so we can take these equations, and as you imagine, there's a spherically symmetric state where nothing happens. They're not going to move around. So that's a state, and then you can take that to do a stability analysis. Here we've defined a Pecklin number, which incorporates this flux and this mobility coefficient, the radius of the droplet, and the diffusivity of the chemical species that we have. And here sigma is not the stress, that's the growth rate, that's in the linear stability analysis. And if you do analysis, what you see is, depending on the Pecklin number, initially it's 0. So when this is weak, you can imagine that the flux you're generating is quite small, then you're only going to be staying in this state. And as you increase Pecklin number, the growth rate becomes positive. So there's an instability that develops. And on the right side here, this is a full nonlinear simulation of the PD system we looked at in the previous slide. So we're looking at two things here. One is the velocity of the droplet. The other is the chemical concentration around the surface of the droplet. So that's a function of the angular degrees of freedom. What you see here is, again, a time-dependent simulation. So initially, you start from a spherically symmetric state where there's no motion at all. As we evolve the equations, what you see is when you have flow, you develop this asymmetric gradient that's self-sustained, and then you're going to reach a long-time state where they self-propel. So that's another interesting system of active particles. Okay, so so you can then characterize sort of the long-time swim speed of these droplets, which are typically, different paper.
Same authors.
Same authors, but that's right. You know that? Okay.
So it's
Okay. So you can then characterize the velocity you're getting. This is non-dimensionalized as a function of the Pecklin number. Again, if the sort of the flux you're generating is very small, then you're not gonna go anywhere. And the critical Pecklin number or critical flux you need is somewhere around 4. And as you increase the Pecklin number, you can get a non-zero velocity out of these droplets. Okay. So that is interesting. And then what you can do with these systems is you can have more than one chemical species that are present here. And you can design these droplets that some of them, for example, you have chemical species A and B, whereas this droplet likes A, so it's chemoattractant to one species, but maybe repulsive to the other one. And then you have chemotactic interactions among particles. So what happens here is you're going to see an effective predator-prey interaction. So this guy here is going to produce some kind of chemical that this particle likes. So it's trying to chase that particle. But this particle doesn't like the chemical that this guy is producing, so he's trying to escape. But their mobility is different. So eventually it's going to be caught by the predator here, and then they're stuck there because this guy is attracted to that guy. Okay? And then you have more particles. They can self-assemble due to these chemical interactions and then form these molecule-like structures made of droplets.
Still waiting like for them.
Is there? Yeah. So the point is you can design different systems. You have different kinds of interaction. This is one way to implement, attractive or repulsive interaction in this kind of droplet systems. And the other thing is these droplets are sometimes fairly large. So if you look at it, it's about, 100 microns in diameter. So it's larger, slightly larger than bacteria that we're looking at, but still within the Stokes regime that we talked about before. Okay? So you can have many of these guys, and then if they can form molecules, they can form large-scale structures if they're more dense. And you can do a simulation, like a Brownian dynamics simulation, by sort of modeling, representing their chemotactic interactions, and then you can form these. In this case, this molecule is turning around in the system. So we can think about how to model these systems in a simple way. For example, you have a particle that produces some sort of chemical, right? So there's a gradient radiating outside. For example, there's a surface reaction from converting S1 to S2. You have other particles that likes this particle, and then you have a chemotactic gradient, which means they get a velocity when they sense a gradient in their space, and then you have a mobility coefficient. You may also have alignment to the gradient of the chemical. And you can have more complex networks of reactions as the one we talked about before. Say you have two types of particles. One is producing some chemical, but that serves as a reactant for the other guy. And then you can also have reactions happen in the bulk fluid, which may convert this back to S1 as well through some other chemical reaction mechanism. So you can allow you to maintain a non-equilibrium steady state. So you can model these systems, and let's briefly look at how one can incorporate this into the Brownian dynamics simulations. Okay, give me 1 sec.
Yes. This one.
I didn't bother to write out these equations in the slides. That's why we're looking at this PDF here. So let's think about particles or droplets. That is what?
Yeah.
Particles that has some sort of surface reaction, or they're just trying to produce some chemical which comes out of these droplets into the fluid. Okay, so this is describing the conservation law for species J. So it's a chemical concentration, the concentration of species J in the fluid, and it follows diffusion equation, advection diffusion equation. So that's their diffusivity. And what are these things here? These are just, because these are produced by these particles, so we model them as some fluxes and then localized at the particle position, that's capital X, and there may also be bulk reactions that are happening entirely throughout the fluid. So there's a general representation of that. Okay? And then you just write down your particle equation of motion. That's the capital X, so that's the velocity of the particle, labeled IK. So there are different particles that are labeled differently. They have their self-propelled velocity. Because they're active due to these chemical concentrations, you can model them. You can model them as dependent on the concentration. These relations you can get from, say, experimental measurements. And then these are the chemotactic responses. So I sense a chemical gradient, I have a response coefficient that gives me another kind of velocity. And I have, what is this one? Okay. See, I cannot remember. This is my own paper, but I cannot remember this. So this is the interaction. So particles, again, moving around. This is a non-dilute system, typically. So you have particle interactions that happen. So here, that's represented by this guy. And then you have other terms here. And then, again, these are your regular Brownian motion. That's the last term, the velocity due to the Brownian motion. And then, again, similarly, your angular orientation vector follows similar kind of structures. You have Brownian part, and you may have alignment, chemotactic alignment in the system, and that is it. Okay, so you solve this coupled to the transport equation. So in this case, you can assume that usually the velocity you're getting from these kind of reactions is quite small. So you can, to first degree, approximate these equations as diffusion equation. You see here, I didn't even have the advection. I've already neglected that. So that's just diffusion equation with some sources and sinks, which we can solve with fundamental solutions. So actually, we don't have to solve this equation. We have analytical formulas for it, and then you just evolve the Brownian dynamics over time, and that allows you to simulate these systems in the simplest way possible. And that's what we've seen here, essentially, in this in the slides that I just showed you here.
This.
Okay, so then you can do this kind of simulation fairly easily. Any comments about this? Okay, good. And then you can have, say, here's an example where these question. Question? No? Good. Okay. So then you can, here we have sort of these two species, and by changing the coefficient of the chemical attraction or repulsion, you can form different structures. For example, in this case, these particles just, they like their own product, so they're great aggregate among themselves. Whereas in another case, this yellowish particle is attracted to the blue one, so but the yellow guys are more active, they swim faster, so they will encapsulate these blue particles. You can get all kinds of different structures depending on these parameters. We have a lot of parameters that one can tune. Question?
Reporters.
Okay, yes, you want some materials where you want some structures. The question is, how do you assemble that structure? And is there a path from these kind of interactions that gives you that? Yeah.
Yeah.
Okay. This question.
Marcus.
Simulations or modeling. You just need to.
So in that, we can do this kind of being fed fluctuations.
You can do the same modeling. Yeah. Since you have a mechanical constraint among particles you have to take into account. But other than that, it wouldn't be different from how we approach these systems here. Yeah. So okay, so we're done with this kind of system. And now let's think about some other interesting cases. There's still droplets, but this is a composite system. The droplet is, notice it's quite large, okay? This is a scale length bar of 2 millimeters. So it's a large droplet. And there are things inside the droplet. They're just bacteria. Okay, so it's E. coli solution in a droplet. Outside is a different solvent where there's no bacteria, okay? Everyone's clear about the setup? So you encapsulate active particles in a droplet. Guess what happens? And what happens then when they go to the boundary?
We take some deal from this.
No, they don't. They are E. coli. They're not. Take what? No, they don't like the things outside.
Failed this
Sorry?
tree. I lost show as the kind of their tools.
So I have a droplet, okay? A droplet of water and bacteria. So bacteria is inside the droplet with water, and the outside is a different solvent, which is not getting into the droplet, okay? They don't like each other. So the question is, we have bacteria in the droplet, and what happens to the droplet?
What is the
To the droplet.
it is lazy with it because
If it's
flexible the
droplet is flexible, that's the definition of a droplet. Droplet has to be flexible.
like force the problem
So what happens to the shape of the droplet?
changing is
All right?
shape.
Yeah, so the shape will fluctuate, right? Because these guys get to the boundary, they're pushing, but the boundary is soft.
What question is never effect we do that says
What?
it's a back together the zero be busted but
There will be a bunch of bacteria.
He said that is can there be like it's cannot be done by one bacteria. We are pushing the
Yeah, they can destroy their, yeah. One cannot normally, but if they are very active, they could, yeah. Destroy the droplet. The other is the droplet may move around. What else can happen? Okay, that's good. We already got some good answers. So let's watch this video first before we move on. So the shape is just fluctuating. In this case, not very noticeable because the activity is low here. You can still see shape is deforming right over time. And what we're seeing here at the bottom is the velocity. So on the left, that's the velocity field inside the droplet. And we now understand why there's velocity. That's generated by the bacteria in there, right? For that, we can easily model with the mean field we talked about before inside the droplet. And this is the vorticity, which is the curl of the velocity. But nonetheless, they're just fluctuating. Now you see there's no particular pattern to that. Question? Okay. Now we increase activity
Yeah.
from the previous, and that's what we're doing here. Of course, you see one thing right away is the shape changes much more than before because it's now more active. And the other thing is, you see, this is it actually goes into a vortex regime. It's spinning.
Just saying the bacteria will go